What is EX? What is EX? Excitation. So, what is the excitation wavelength? What is the dichroic mirror? DM stands for dichroic mirror, or some nomenclature will say BS, beam splitter. And the third thing that you will see is EM, emission filter. EM stands for emission. So, a typical GFP filter or Alexa Fluor 488 filter shows a specs like something like this. Something like this, something like this, and sometimes something like LP 510 nanometer or 510 to 550 nanometer BP or 525 plus minus 50 nanometer. Sure. I'm coming to that. Okay. So, these are all wavelengths. Nanometer means wavelengths, lambda. Correct? So, here, when you say waveguide, it means that right starting from 400 nanometer up till 700 or 650 nanometer, you have the light available. Correct? And then, this 460 to 490, if you look at the visible spectrum, what color it denotes? What color in this visible spectrum it will denote? Okay, let's say blue. Very good. So it means that whatever white light is coming over here, only this particular wavelength or range of wavelength of light is allowed to pass. So it means that your white light gets converted to a blue light. It means that the blue light comes and enters the filter cube, and here you have a dichroic which is having a wavelength of what? So this functionality of this dichroic is like a beam splitter. It splits the wavelength in two components. Two component means one lower than this value and one higher than this value, simple. So lower than this value means what? The same wavelength. Means the same blue light. Your dichroic is inclined at 45 degree means light will go 90 degree below. So whatever light came in 90 degree, you have the objective and then you have your sample. That is how you have seen a fluorescence microscope. So far so good. So now the blue light excites your sample. Clear? So far? Any issue over here up till here? So now that you have excited your sample and wherever you have the fluorophore or the protein or the tag, it will excite that. Some work function loss, energy loss will happen, some absorption will happen, and the remaining energy means lower energy than the excitation or lower energy than the incident light. So some work function loss and the remaining energy will transmit back or reflected back. So when I say lower energy means higher wavelength, lower wavelength. Higher energy means lower wavelength, higher wavelength. So energy is higher, energy is lower, which will have a wavelength higher. E is equal to hc by lambda, simple. That's the formula, right? No? Okay. So the formula is very simple over here. hc by lambda. Energy is inversely proportional to wavelength. So lower the wavelength, better the energy, higher the wavelength, lower the energy. Simple as that. So far so good. So it means that you excite your sample at a higher energy, lower wavelength, and whatever energy or remaining energy means lower energy means higher wavelength will now pass after this 500 nanometer. So the splitting is lesser than 500, higher than 500. Lesser than 500 means this, 460 to 490 means excitation light. Anything higher than 500 will go into the emission side, that is your emitted signal, or lower energy or higher wavelength. And typically, if I say that you have this, means anything from 510 to 550 nanometer is only allowed to pass, because this filter will have that specification. Means now when I say 510 to 550 means what color now? So it means that you excite your sample with what color? Blue light. The emission that you collect in your eyes is what? Green. Got the concept? So unless and until you have a cube that has all these mirrors, you will not be able to excite and collect the emission. Is the basic concept of a fluorescence microscope. It's a pseudo color. That's altogether a different thing. When you see the sample through your eyes, it's the color, natural color that will be coming out of this emission. But when you try to image it through a camera, your camera can be a color camera, your camera can be a monochrome camera. So when you have a camera which is monochrome, which is slightly more sensitive as compared to a color camera, you will try to give a pseudo color. And that pseudo color, your choice. Okay, I have visualized green. Why I should give a green? I will give a yellow color. I will give a magenta color. It's my choice. Yeah. So the concept here is very simple. You excite. So now excitation is blue, emission collected is green. If you have a DAPI filter, anybody have seen or known about DAPI? Nuclear staining. Excitation is violet, emission is blue. If you have a Psi3 fluorophore or a protein or a organic dye, excitation is with a green color, emission is with a red color. And if you have a Psi5 or a far red filter, then excitation is by red, emission is far red. Far red you cannot visualize with your eyes. So forget about visualizing Psi5 dyes with your eyes, but the camera can acquire that because camera spectral detection capability is up till 1000 nanometer or 1100 nanometer. So with this concept, if you understand this, it becomes easier for you to understand the basic advanced level of microscopy, what we call as confocal microscopy. So what is confocal microscopy now? Now that you have understood basic fluorescence, why you need a fluorescence microscope? To excite your samples at different excitation and collect emission and the difference in between the excitation and emission, this gap is what you call as, there is a nomenclature to that. Any idea? The difference in between the excitation and the emission is what you call a Stokes shift. Stokes shift. And this is the basis onto which any fluorescence microscopy function. Forget about brand, forget about which model. Any basic fluorescence microscope concept will be based on this aspect. A slight modification and you get into confocal. So first of all, let's understand why confocal. What is the difference between regular fluorescence microscope and a confocal? Anybody? Any idea on the confocal till now? No. Okay, very good. So, forget about that. So it means that if you have a fluorescence microscope, you excite your sample. Your samples can be monolayer, your samples can be multi-layer. Matlab, most of your samples will have a certain thickness, depth of penetration. And mostly these samples are transparent, means light is able to pass through. So it means that you don't only collect single plane information, you also collect information in the Z. Because you are exciting the complete volume, you are collecting emission from the whole volume. And typically when you try to visualize that complete emission through your eyes, you see a lot of useful information, but you also see a lot of haze information. What is haze information? Unwanted blur information, out of focus information, which you can't focus properly or which you can't see properly. And it is coming because of the scattering of the light in that tissue or in that volume while you are doing a depth of penetration imaging or while you are doing your in-depth fluorescence imaging. So you never know that, okay, I have got a good emission signal, but it's a lot of haze. I am hardly able to understand which image is coming from which focal plane. And what is the focal plane? The best focus. The best focus at which you get the ideal information or the in-focused information. So now typically when it turns out that, okay, so it means that with a fluorescent microscope, unless and until my sample is very fine or very thin, I will always get that haze information. It's never possible to generate an image free of any haze. When we come to a confocal microscope, it does some magic. Your confocal is nothing but an extension of a fluorescence microscope. आप इतना समझ लो अभी. It's nothing but some additional properties, some additional aspects that gets into the system that converts a fluorescence microscope to a confocal. And every system will come with its advantages, and it also will come with its own disadvantages. In a confocal, for every advantage, you will have a disadvantage. So what does then confocal microscopy look like? So now this white light, so typically what source? Any idea? It can be a mercury illumination, it can be a xenon illumination, it can be a metal halide illumination. So these are different type of white light sources that can be made available for fluorescence. But then this is a white light means it's a non-coherent source of light. Anybody understands coherent, non-coherent? Any idea? No. Okay. Anything that changes phase or there is a phase change, because you see so much of wavelengths, there will be some phase change. So these are non-coherent source of light. When I say coherent means one single phase, one single frequency, one single wavelength at which it will excite your sample. So your lasers, so on a confocal, what changes is this white light gets converted to a laser source. What is laser? Any idea? Have you heard about lasers? What is the full form of laser? There is a full form, come on. Laser amplification by, any idea? Standard emission and depletion, LASER. Okay, so for your understanding, just understand that it is a coherent source of light and it excites, or it has one particular wavelength to it. To make it very simple, that is how it looks like. And then what happens is, so then you have a laser. So it means that you have now got a laser which is exciting at a particular wavelength, say 488 nanometer, and then you have an optical fiber, the light source or optical fiber through which the light comes. And now if you are having a source which is exciting at a particular wavelength, unlike a fluorescence microscope where you have a white light and then you have an excitation mirror. So here, do I need an excitation mirror? Because I am already having a wavelength or a laser exciting at a particular wavelength. So it means that there is no concept of excitation mirror or excitation wavelength to be available onto a confocal. What is the next thing that was there in the fluorescence microscope? Inclined mirror dichroic or the beam splitter. So the first component that will come onto a confocal is your beam splitter, or what you call as a nomenclature we say MBS, main beam splitter, for instance. Clear? So now laser light, deflected, excited, objective, and then you have your sample. Same, exciting all the complete volume, all the Z planes, and then some work function loss, remaining energy comes over here, and now whatever is higher than the excitation or the MBS, 488 will go into the emission side. Now in the emission side, you have a magical wheel, what we call as a pinhole. A pinhole. And this is a variable pinhole. This pinhole is variable, governed by one formula called 1 AU or 1 ARI unit, 0.61 lambda by Na. ये खाली फॉर्मूला याद रखना आप लोग, बाकी सब कुछ इजी है. 0.61 लैम्डा बाय Na. 0.61 फिक्स फैक्टर. लैम्डा क्या है? एक्साइटेशन वेवलेंथ बोल लो या फिर एक्साइटेशन प्लस एमिशन डिवाइड बाय 2, जो बोलना है बोल लो आप. Na क्या है? Numerical aperture. So what is numerical aperture now? N into sin theta. Very good. So what is that? What is that? So NA is N into sine alpha or sine theta. What is this theta? So it means that every objective opening, whatever is that, this is 2 alpha or 2 theta. Half of that becomes your theta over here. Okay? So what is this N? What is this N? This is the refractive index of the medium. So if you have air, what is the refractive index? If you have air, what is the refractive index? 1. Very good. If it is water, what is the refractive index? 1.313. Very good. If it is oil, what is the refractive index? 1.515. 1.515. Very good. So you guys have understood quite a lot. Okay. So it means that the NA is nothing but the light accepting capability of an objective. The more the NA, the better the light collection capability, or the better the angle. So the more the angle, more the photon collection or the light collection. So whenever we talk about confocal, higher the NA, better the resolution, lower the NA, bad the resolution. What is resolution? How do you define resolution? Tell me, ma'am. Okay, in layman terms, yes, pretty nice. So if I say 2 dots, and I can image it as 2 dots, yes, it is resolved. But if it is 2 dots like this, can you resolve it as 2 image? No. This 2 dots I can image and resolve it as 2 dots or 2 images until this point. The time it gets overlapped, you go beyond the diffraction limit, or there is no, you cannot resolve it. So up till here it is resolvable. Beyond this it is not resolvable. So the ability of a system to resolve 2 dot as 2 or 2 point as 2 points is what you define as resolution. Okay? So it means that if I say 2 dots over here, try to image it, this is how I will get an image. Right or wrong? If I have 2 dots like this, and then this is how it will be. But if I have 2 dots one above the other, then I cannot resolve it as 2. It will look at is one single dot. So this is limit of resolution. This is still, I can resolve much better than this, and this is beyond the resolution, or what we call as beyond the diffraction limit. Okay, so the idea here is this NA is responsible for giving you this number, and this lambda is responsible for giving you this number. So this will allow, so what happens is you are now collecting emission from the complete focal plane, right or wrong? But with this pinhole in the conjugate focal plane, what we call as, the conjugate in the emission side, it allows only the in-focused information to pass through this pinhole. Whereas in a regular fluorescence microscope, what happens? You excite the whole volume, you collect emission out of the whole volume, and you don't know that which information from which focal plane. But in a confocal, when you have a pinhole available in the conjugate focal plane and with a pinhole opening of 1 AU, it allows only a single plane information or the in-focused information or the conjugate focal plane information only to allow to pass through that. Rest, all the other will be obstructed. It means that you clearly get a information from a single plane without any background from the above and below focal plane. So far so good? If I try to move the sample up and down, means I change the focus of my sample, means I want to image at a different focal plane. So it means that another single XY plane image will be acquired without any information from the above and below focal plane. So it means that I can start my Z imaging, or I can start my XY imaging at different focal plane to collect single XY images at different Z position. Yes. I'm coming to that. So right now it's only 2D versus different positions of Z. And when you combine all this 2D images at different Z position into one, you can create a volumetric data and what you call as a 3D resolved structure or a 3D structure. And totally in focus information in the whole field of view. Okay, so let's go a little bit further. I think you guys are well known now. Forget about this. So typically when we talk about imaging, when we talk about fluorescence, we don't talk about only fixed sample, we talk about live samples. We talk about samples that are changing fluorescence intensity over time, or there might be samples that are quite thicker and you want to do a Z stack. So right starting from 2D imaging to 6D imaging. When I say 2D imaging means X by Y image, single plane. When I say 3D means X, Y versus Z. And then you can create a volumetric rendered data. It can be 2D versus time, a time lapse if it is a live sample. It is showing some abrupt changes, some vesicle transports are happening. I can record it, I can image it as a 2D versus time or a time lapse image. If it is multiple fluorophore, because in one sample I can have green tag, I can have a red tag, I can have a nuclear staining which is a blue tag, so multiple wavelength. So X, Y versus time versus lambda. X, Y with Z, with time, with lambda. So you see, from 2D I came to 3D, then it became 4D, X, Y, Z versus time. It became 5D, X, Y, Z versus time versus lambda. It went to 6D, X, Y, Z versus time versus lambda versus multiple position. If I have a scanning stage or a motorized stage where I can have multiple location, multiple position of the sample, so I can do right starting from 2D imaging up to 6D imaging. Most of your samples will be depth perception sample, means there will be some depth you would like to understand how the vesicles, or suppose somebody is working with something that shows changes in the Z volume. So unless and until you acquire the Z over a period of time, you will not be able to understand that volume. And so the basic idea of a confocal is not only for image acquisition from 2D to 6D, but also image quantification. What do you understand by quantification? How do you quantify? First of all, what is quantification? Quantification of data means how the signal is behaving. Whether it is, weak signal, whether it is strong signal, at two different locations, is there a change in the intensity of the signals? So that is what you call as quantification. Trying to understand that, okay, if I have taken 10 region of interest or 10 cells, how these cell intensities are minus the background. And this can only be done with a confocal, or this can be done pretty well with a confocal, because this pinhole at one area unit doesn't allow any unwanted light to go to the detector. It allows only a single plane information to go to the detector, for which you know the signal and you also understand the background. Right? So you can quantify your signal pretty well when we talk about confocal image, when we talk about depth of penetration with time, or XY versus time, or just XYZ image. So now, if I try to so anything that you can expect out of a fluorescence microscope, you can expect the same thing with a confocal. With the exception that you can resolve structures, you can quantify structures, you can understand colocalization. Anybody understands colocalization during the fluorescence microscopy? Anything with respect to colocalization? So at one location, if you have two proteins or multiple proteins, and if they are quite close to each other or they are interacting closely to each other, you say it's getting colocalized. So when we talk about colocalization and when we talk about a fluorescence microscope, if you say that you have a green channel fluorescence and you have a red channel fluorescence, what is the combination of green and red? What is the color that you will get? Yellow. So seeing a yellow color, oh wow, this is colocalizing. But you never know that what focal plane, or it is the haze information that is giving me that color. With a confocal, you can do quantitative colocalization, because the same sample will be acquired at the same focal plane without any other out of haze information, you will be able to quantify the intensity values of green minus the background, intensity value of red without the background, and you can easily quantify the two signals, and you can understand the co-localization aspect in terms of numbers. So your confocal is not for doing pretty images only, it's for data analysis also, it's for acquiring data with numbers. And what is important is not just beautiful images, but the numbers that are associated with it. Okay? But what are the losses that you have in a confocal? Now that you see the pinhole is one area unit, means closely tight opening, allowing only a single focal plane image. So it means that, and if I try to open this pinhole completely, then it becomes a regular fluorescence microscope. That's the major difference in between, because this is the pinhole and the laser light, that's the only two difference majorly. So it means that unless and until your sample is good enough, less prone to bleaching, it has to be stable, then only you can expect to acquire your data with a confocal. But if your sample is dull, it is bleaching fast, if it is not stable, you will not be able to acquire your data, because you have lasers that is more penetrative in terms of energy, more higher energy as compared to a non-coherent white light, and you will bleach the pinhole in the conjugate focal plane is at one area unit, and you ensure that you collect information from a single focal plane. Good. So now, if you see these two images, you see the first image over here, what is this? White field. If you see over here, you have got the confocal scanning. You see what is the difference in between these two, looking at these two, or if you look at these two images, you can feel the difference. What is there? What is this image over here? White field fluorescence. A normal conventional fluorescence microscopy, where you see signal but lot of haze also. Lot of unwanted information in the background which you can't resolve, or you never know that which information from which focal plane, right or wrong. Why? Because you excite the whole volume and you collect emission out of the complete volume, which means that in focus information plus out of focus information, everything is collected. What happens in a confocal? You excite the complete volume, you collect emission out of the whole volume, but because of the pinhole in the conjugate focal plane, you acquire or you collect only the in-focused information or the conjugate plane information only, and then this is not a single plane image, but a combination of single images at different Z position and a MIP, maximum intensity projection, to give you a totally in-focused image in the whole field of view. And here you can clearly say that, okay, the background is quite less, I can easily quantify my signal of the background, I can easily quantify my signal, and then I can subtract the intensity of the brighter minus the background to get the actual intensity or the correct intensity of my single actin filament or single neuron or single nucleus structure like this. And that is where the advantage of confocal comes in. Definitely, this is a much better image as compared to this, but this image has more information than this in terms of haze information, in terms of blur information, which you can't quantify. So, but then here you get useful information. That is what is the idea. Although you are compromising on the light collection, but what you are collecting is only useful information, garbage is out. So far clear? Good. So now, this is how you try to do a Z-stack, and that's the answer to a question. How do you do a Z-stack and how you collect a 3D volumetric? Every single plane you acquire an image and you collect all these informations to create a volumetric rendered data. Or like the MIP which I mentioned to you right now. And the basic advantage is in-focused information at every focal plane. You don't have any haze information from the above and below focal plane that you are acquiring over here. When it comes to microscopy, I mentioned about 6D microscopy, but it's not only about 6D. Depending upon sample to sample, you might talk about getting a better resolution, getting a better sensitivity, getting a better speed, getting a better depth of penetration. and it all depend upon sample to sample. So what is resolution? Somebody defined very clearly. What is sensitivity? How do you say sensitivity? I mean, sensitivity, how do you define sensitivity? The ability to detect the faintest of change or the slightest of change. Good. So here when we talk about confocal, we talk about low light level signal, we talk about fluorophore imaging, and whenever we talk about fluorescence imaging, we talk about dark background. We talk about the dark room image because I don't want to capture the stray light. So when we talk about sensitivity means capability of the system to acquire low light level signals. Because what matters at the end of the day is a good image. When you say good image means what? Optimal signal, minimal noise. When you say about an image, you always talk about S by N, signal to noise. Optimal signal, minimal noise. Then only your S by N factor will be maximal. If the noise is more, signal is also more, then S by N reduces. Or if the signal is constant, noise increases, then your S by N reduces. So the idea is to detect the faintest of information, I need to have a highly sensitive system or a highly sensitive detector or a highly sensitive confocal. And when I say a high sensitive detector or a high sensitive system means the minimal amount of signal that can be acquired or that can be captured without the background or without the noise. Simple. Whether it is camera, whether it is your eyes, when you say that my eyes are more sensitive than yours, what does it mean? Does it mean that I can look much more finer, much more cleaner, much more? More? Okay. Fine. So when I say that, okay, I have a camera and I can use this camera to detect the stars in the night. So what does it mean? Or I have a camera, I cannot image in the dark, but in the bright I can image it pretty well because my camera is not very sensitive. Or I cannot detect low light. So it's all about the faintest information with the minimal amount of noise that can be acquired. And the same thing applies into a confocal. On a confocal, what is a detector? Your eyes? No. No, it's not the camera. So these are special detectors, what you call as PMTs, photomultiplier tubes, or some refined detectors like what we call as GASP, gallium arsenide phosphate detectors. Or so it all depends upon what type of detectors you are using. Good. And these detectors are available into a confocal because when we talk about a confocal, we don't talk about imaging parallelly a single plane, means excitation falling on a single plane and emission collected parallelly out of that single plane. Confocal is, it's called as laser scanning microscope. So what do you mean by laser scanning microscope? Means it is illuminating an image or it is illuminating point by point to create an image. Like a EM, SEM, scanning electron microscope, where you have a scanner, you have an electron source. So here you have photon source, you excite one point, you move to the second point, you excite that point, move to the third point, excite that point, till the time all these 5 and 2 points. If you say image is 5 and 2 by 5 and 2, then you have to illuminate each point. First horizontally, then come to the second line, again horizontally all the points, then come to the third line, again horizontally all the points. So
what do you mean by noise?
Noise. Yeah. Noise is, okay, so if you get this whole room dark, and then, okay, forget about that. So, okay, if your signal is low, and then you try to increase the intensity by applying a better laser power or by increasing the detector gain or by applying a better exposure time to a camera. So the more the exposure time, you generate better signal, but the background also gets lifted up. Noise is nothing but signal coming from the background. Background means where there is no sample. So if you are trying to increase the gain or of say exposure time or the detector gain by applying a voltage, the higher the voltage, higher the signal, but it also improves the noise. So signal is improving, but noise is also improving, so your net effective signal to noise Whereas if the signal is optimal, so the signal is bright, I will not increase the exposure time by a certain value. And that value is nothing but my optimal signal with the minimal of noise or the minimal of background signal. So if I say that, okay, you have an image, you want to image, and my image is an 8-bit image. You know 8-bit, 10-bit, 12-bit? Anybody interested in imaging over here? Camera-based imaging, photography? You understand 8-bit image? What is 8-bit image, sir? How many grayscale values? Darji, tusi bolo. 8-bit image, how many grayscale values? Okay, so if it is 8-bit, grayscale values 2 to the power 8. How much is 2 to the power 8? 256. So on a scale of 0 to 255, whatever is 255 is brightest, whatever is 0 is darkest. Clear? So whenever you acquire an image, you will always define your image with respect to bit depth and grayscale values. And these 0 to 255 are nothing but pixel intensities or grayscale values only. So anything, suppose you have an image 8-bit, matlab maximum grayscale value is from 0 to 255, right or wrong? Anything more than 255 is what? Not 8-bit. It goes beyond the scale, which you cannot quantify, which you cannot resolve. Anything less than 0, can you quantify? So if you have a scale which is 0 to 6 inch, 6 inch scale, and suppose your sir says, okay, show me a measurement 7.5 inch, can you do that? No, because your scale is not compatible to measure 7.5 inch. But if somebody says that, okay, with a 6 inch scale, show me a measurement 4.5 inches, you can easily do that because it's within that dynamic range of your scale. Similarly, if you talk about a 10-bit image, how many grayscale values? 2 to the power 10. How much 2 to the power 10? 1024. 2 to the power 10 is nothing but 1024. So on a scale of 0 to 1023, whatever is 1023 is brightest and whatever is 0 is darkest. And suppose on this scale, if my intensity value or grayscale value is 550, your grayscale value is 600, you are more whiter than me. Simple as that. So that is how you do a comparison. And that is how you quantify intensities in between two different positions. So in a cell or in a field of view, suppose you have acquired a fluorescence image, you have 10 cells into that field of view, and each 10 cell will have different grayscale value or different efficiency or different quantum yields depending upon the structure or depending upon the protein, and that is how you differentiate in between the yields, or in terms of quantification you will say, okay, this is the most brightest sample, this is the more dullest, this is in between the brightest and the dullest, because I know the intensity and I know what is my background. And that is what quantification means in terms of image quantification. And this you cannot be done with a regular fluorescence microscope because of the haze information, because of the blur information. That can only happen with respect to the confocal microscope. Good? Chaliye. So now try to see some beautiful images. Now you see, any idea what are these structures? What does it look like? What is this structure? This is a brain, Drosophila brain. And you see two fluorophores over here, two colors over here. What are the two colors? One is green, one is red. So definitely one with one particular wavelength, the second with the other wavelength. So it's a two-color image, but not a single plane image, because you see everything is getting focused over here. So it's a XY with Z and then a MIP, maximum intensity projection, to give you in focused information in the whole field of view. This also is the same thing over here. This is a Drosophila embryo, a portion of a Drosophila. And the GFP, which is the green one. And then this is again not a single plane image, but a totally in-focused image after doing XY images at different focal plane and then doing a MIP combined together. And this is easily resolvable, easily quantifiable, because you don't have any unwanted structure, you don't have any haze information, you don't have much of background, and you are able to easily quantify how much of intensity of this minus how much is the background, or the comparison of the intensity of the green with respect to the red over here. Okay? Now, see this over here. This is also a spinal nucleus dies, and you can see the depth, which means that it's not a single plane image, but we have done a Z stack, multiple fluorophore, and then did a MIP over here. Images over here. This is also a mouse brain, and you can see the neurons, and you can see the oligodendrites clearly over here. You see the fascinating world of plant samples. Anybody understanding bio, botany, plants? Okay. What type of plants? Arabidopsis. You understand Arabidopsis? Okay. This is nothing but a Arabidopsis leaf, and you can see what are these structures? Any idea? At least I don't have an idea because I am not from the bio background. What are these? Stomata. Very nice. And you see some red, some green. So these reds are nothing but the guard cells, the cell walls. You know, plant has a lot of autofluorescence. Full of autofluorescence, chlorophyll, chloroplast autofluorescence. If you want to acquire that and give a separate pseudocolor or separate that autofluorescence with respect to a green fluorescence, confocals are the ideal choice. You see over here, how many colors over here? Total. Can you know this? Total of 6 colors. And you see 6 fluorophores on a single sample. So you can see multi-channel, multi-color fluorescence imaging. Forget about one single fluorophore. You can talk about multi-color, multi-fluorescence imaging at any point of time. And bleed through free images that you can generate with a confocal with quantification of your data. You talk about depth, much better depth of penetration. Why? Because you have the pinhole, it ensures that you collect information from a single plane, laser being a coherent source of light, more penetrative, more energy, I can image much further deeper in my sample, I can generate photons from the depth, and I can create a volumetric rendered data out of it, or I can create a 3D reconstructed data out of it. See this image, these are all plant samples. And the basic idea of plant samples to show is not only the image being beautiful, but also the challenges that you face. If you just try to take a stem section or a root section, just cut it from the garden and put it on a glass slide with cover slip, you will hardly be able to see anything. Because it's full of structures that are quite scattering when you go deeper. But then if you do a little bit of better optical sectioning or a little bit of better slicing with a blade even, you will be able to get to the guard cells. You will be able to get to the chloroplast cell wall or the chlorophyll cell wall autofluorescence. And you will be able to get these structures pretty well. So that is what is the scenario. When it comes to live sample, now you see over here. So it means that not only fixed sample. Now you see this is a live sample over here. And this is again a Drosophila embryo. And you see the data, 2000 images, 20 number of Z-stack, 100 number of time point. So at each time point, 20 number of Z-stacks. And then all these 20 Z-stacks getting combined into one to give you a MIP. And this is where you can see the opening and closing. And you can see the intensity has not gone bad even after 20 minutes of imaging. And laser falling continuously on the sample. If you see this over here, time data is changed. From 20 minutes, it has gone to 3.5 hours. And this is 8,000 image. So for 8,000 image, again 20 number of Z-stacks and 400 time points. For all this 400 time point, the laser was there, the laser was activated, the laser was falling on the sample, the detector was collecting the emission, and the sample was stable enough and the laser was optimal power so that there was no bleaching happening on the. You understand what is bleaching? Fading of the fluorescence, because the illumination can be at a slightly higher power and that will induce photobleaching phototoxicity. And when it comes to live sample, photobleaching phototoxicity is much more profound as compared to fixed sample, because you have the concept of adding some anti-fade and no anti-fade with respect to live sample. Live samples, you work with proteins, GFP or variants of GFP. Fixed sample, you can work with any of the organic dyes where you can use some anti-fades. But these anti-fades can only work with fixed sample, it cannot work with live sample. If you go further over here, now you see 2 hours imaging, and this is a Drosophila, zebrafish embryo. Over here you can see the vesicle transport, and you can see the GFP, and this is basically the cell division over here. You see over here. So where was 20 minutes? Where was 3.5 hours? Where was 2 hours? Where is this in minutes? Where is this in seconds? So you see the time resolution, time stamp. From seconds to minutes to hours to days and even to milliseconds. And it all depends upon what type of functionality or what type of samples you are interested in imaging. Somebody will show quite abrupt changes. Somebody will show some transports really fast, some actions really fast that you would like to acquire. So how do you need to acquire this is trying to do a little bit of play with the confocal system, trying to set the adjustment in such a way so that it can do fast imaging also. Apart from being suitable for fixed sample, I want that system to be active and good for live samples also. So it's all the instrumentation part that you have to play a bit and then set the parameters in such a way or set the experimental setup in such a way so that all this type of experimental data can be acquired without much of limitations. Now this is all single protein. Now you see dual protein over here or dual color. Now you see now this is a blood vessel. This is a Drosophila fish imaging. You see, you can see the red blood flow and you can see the green structure, and this is a time series data in seconds, like 20 seconds imaging, and it's a fast imaging. If you see this particular image, this is again a multiple color, two color protein, so GFP M cherry, X, Y, Z versus time, and then you can see the dividing cell over here, you can see here, one cell dividing into two over here. And for all this thing to happen onto a microscope, ultimately, a confocal is nothing but a microscope only. To happen this onto a microscope, you need to have precise controlled environment on a system. And when you say controlled environment means what? I need to make my cells happier on the microscope stage. So it means that CO2, temperature, humidity, in terms of hypoxia experiment, O2 control, everything has to be intact. So it means that I have to build a mini incubation system on a microscope to ensure that my samples are quite well for long-term imaging because my sample is out of the CO2 incubator box for that period of time. So I need to create the incubation environment on the microscope. And these all things are now possible. Or it can be done pretty well onto these systems. Good. So you can see this, another image, cardiac myocyte. Anybody understands what is this cardiac myocyte? So this is like fast calcium flux imaging, and this is like 200 images in less than 2 minutes. When you say 200 images in less than 2 minutes, then you need to have a system that is pretty good, pretty fast to acquire this data and collect it with quantifiable signal information. So the same confocal with a bit of modification with respect to experimental protocol and experimental setup, you tend to get images like this. If you see this particular image, you see different Z positions, right? Or different Z. And you can see the vesicle transport at different Z position. And all this again is possible because you have a pinhole available in the conjugate focal plane at one area unit that doesn't allow any of the out of focus information to go to the next plane. So this is X, Y, Z versus time. Clear? Good. So now experiments like this. Anybody understand what is this? Any idea? What is it doing? What is happening over here? Anybody understands FRAP? Fluorescence recovery after photo bleaching? No. So this is nothing but a FRAP experiment. where you tend to bleach a small region and you see the recovery over a period of time, and this recovery is happening because from the neighboring some molecules are getting diffused into the bleached region. So it means that your cells are active, cells are live, and there is a movement or there is a diffusion of the molecules from the unbleached region into the bleached region. So this is what is understood by Frapp. So all this type of experimental studies, if we talk about a confocal, can be done pretty well with quantifiable data, with quantification, and that is where the advantage of confocal microscopy comes in. So till the time it is XY resolution, there is no difference in between a fluorescence and a confocal. The resolution is exactly same, diffraction limited to 20 to 30 nanometer XY. When it comes to Z resolution, there is no concept of Z resolution on a fluorescence because there is no way you get rid of the haze information. So optical sectioning is possible, but precise optical sectioning to generate a Z resolution is not possible. When it comes to confocal, the only advantage that you have is axial resolution, means your Z resolution. XY resolution of a confocal and a fluorescence is exactly the same. What is important for a confocal is your Z resolution. As a thumb rule, Z resolution will be twice as bad as XY resolution. As a thumb rule. Means if the XY resolution is 240, 250 nanometer, your Z resolution will be more than 500 nanometer or close to 500 nanometer. Or typically, if you don't understand this concept, just try to understand that whatever is your laser excitation, your Z resolution is close to that excitation number. Half of that will become your XY resolution. So if it is a 640 nanometer excitation, your Z resolution will be close to 700 or 650, 640. Your XY resolution will be 640 divided by 2. So your XY resolution is always 2 times better as compared to Z resolution. 0.61 lambda by Na is diffraction limited resolution XY. 2 multiplied by that factor. मतलब 1.22 lambda by Na becomes your Z resolution. इतना समझ लो अभी तक यही काफी है. And then with this I would like to end my talk. Yeah, and thanks for not sleeping and thanks for being attentive. Any questions? I am more than happy to answer. Thank you. Any question? Tell me.
So I still did not understand the point of a pinhole.
Okay. So when you talk about, okay, so this is your sample, right, madam? This is your sample, correct? You're exciting the whole volume. But out of this whole volume, one of this plane is your exact focal plane, right or wrong? Your in-focused, precise focal plane. So it means that you have your objective, you have, so the objective is there, the dichroic, and then you have the pinhole in the conjugate focal plane. When you say conjugate focal plane means what? When you say conjugate focal plane means what? Same focal plane. So whatever, as a layman, whatever is the distance of this dichroic from the exact focal plane, the same will be the distance of this dichroic from this pinhole opening. Basic light diagram. पढ़ा है optics पढ़ा है आप लोगों ने? You have studied optics, so you have an object at this position, you have a lamp or a light source at this position, you have a lens at this position. Where is the image formed? Opposite side, inverted, same distance. Same concept. Image of this exact focal plane is falling onto this. And that is only allowed to pass through the detector. If this position is slightly below or slightly above, what will happen? Exact focal plane information will not pass. So you will start getting haze information. Or if this pinhole is more open, what will happen? More Z means lesser optimized Z sectioning information will pass. If I open this pinhole fully, then complete information will pass. So it's important for you to make sure that this works at 1 AU and it's in the conjugate focal plane so that the in-focused information is allowed to pass, because this 1 AU and the in-focused information is what is trying to create a optical sectioned image for you. And different optical sections images at different Z position gives you what you call as a volumetric rendered 3D projection data. That is the reason of having a pinhole and the importance of pinhole into a confocal microscope.
So by information you mean the cell, right?
The information means light collected from this plane in terms of fluorescence intensity. That's it.
Okay. Thank you.
Yep.
Name, place, and then. Good afternoon, sir. I'm Ert Singhal from New Delhi. I wanted to ask, sir, as you told that if we completely open the pinhole, then the confocal microscope will act as a fluorescence microscope. So why do we really need a fluorescence microscope then? Why can't we just build a confocal microscope?
Okay. So how do you know that your sample is good for imaging or bad for imaging? Your lasers cannot withstand your eyes. First thing. So before you get into the confocal mode, you need to visualize your sample in the fluorescence because your tags are fluorescently labeled. So your fluorescence microscope is needed before you get. So I told you confocal is nothing but extension of a fluorescence microscope. Whatever you cannot do with a confocal, you are doing it with, you cannot do with the fluorescence, you are doing it with a confocal. So the first thing, the time you come in the room to set up the system, you put the sample on the microscope, it's a fluorescence microscope. You visualize the sample in the fluorescence mode using the white light, using the the magical filter that I showed you, and then you visualize your sample, you select the region of interest that you want to image, and then you go to the confocal mode with the laser getting excited, because the time the laser starts scanning the sample, your eyes cannot visualize because your eyes will be hurt the time you see the laser into your eyes. So it's a pre-inspection tool, and for every sample you don't need a confocal. So if you have a monolayer cell where you don't have any haze information, why this haze information? Because your sample has a thickness. You cannot visualize or you cannot separate the haziness or the hazy structure at the below with respect to my in-focus information. So when you have a monolayer cell, like a blood smear, you don't need a confocal because you don't have any unwanted haze information. Routine fluorescence microscope will give you an image free of much of haze. Your haze only comes into picture if you have a sample that has a volume, that has a thickness, and invariably 99.9% of the biological sample will have some thickness. Even your yeast, bacteria, fungi, 2.5, 3 micron sample thickness. If it is cell culture, cell line, 8 to 10 micron sample thickness. If it is a tissue, 20 micron, 30 micron, 50 micron, depending upon how best sectioning you have done. If it is a model organism, you are talking about a zebrafish embryo, it can go to 50 micron, 100 micron sample thickness. So the more depth of penetration, the more scattering. And the more optimal it is for a confocal, because the pinhole will ensure that you excite the whole volume, you collect emission out of the whole volume, but you are able to differentiate this single plane information from the other focal plane. And that is where the advantage of confocal comes into picture.